This is the continuation of Chapter 1, and we were looking at this long definition of cancer that was put together by cancer researchers. So we had the abnormal growth of cells, losing this balance between cell proliferation and cell death. And now the second part I wanted to focus on is that this population of cells is going to evolve into one that can invade tissues and even metastasize to distant sites. And so this idea of invasion and metastasis really highlights the difference between cancerous and non-cancerous tumors. Because of course there are two different types of tumors. You have benign tumors that are too many cells, but they're growing in a confined local area. And then you have malignant tumors, which can really spread either locally, regionally, or potentially systemically through the bloodstream, which was what you would consider metastasis. And cancer is referring to any kind of malignant tumor. And there are some weird exceptions. So I showed you a picture of my wife on that first slide, and she had a meningioma, a brain tumor, which is by definition a benign tumor, although in her particular instance it was considered a malignant meningioma, I think just denoting the fact that it was more aggressive and rapidly growing than most typical meningiomas would be. So there's a little bit of a gray area there, but in general, benign is confined to the original tissue, malignant has spread. And you can see this malignancy illustrated on this tissue section here. So here we're looking at colon cancer, colon tissue, and we see normal tissue organization on the left half here. So you have these tubular mucosal epithelial cells, right? You can see them basically packed with vesicles that are going to be excreting mucus. You have this basal layer down here you can see in the gray. And what's going on here on the right, where we have some cancerous cells now, or a tumor, is these excess cells have pushed through that basal layer completely. So if the cancerous cells were confined here, or the excess cells, I should say, were confined up here to the tubular epithelium, we might consider that a benign tumor. But because these have spread and pushed through that basal layer, spread from the original tissue, now we'd consider that a malignancy, a malignant tumor at that point. And I also want to point out there's three main categories of tumors, and I'm sure you've heard some of these terms and you'll encounter them again through the course of the semester. But we have the most common type of cancer or tumor, which are carcinomas, and these are defined as arising from epithelial cells, right, which is a large percentage of the cells in our body, right? The outside of our body is covered with epithelial. All the inside cavities are covered with epithelia as well. Then you have sarcomas, which are the rarest. These are tumors arising from supporting tissues, bone, cartilage, fat, muscle. That's relatively uncommon. And then we have the lymphomas and the leukemias, which are unusual in that they are really considered mostly circulating cancers. So lymphomas are kind of a weird in between. They can grow as solid masses, even though they are cancers of blood cells, whereas the leukemias are truly going to be exclusively circulating blood cells that are malignant. So you might ask, okay, why bother to give these different types of tumors specific names? Tumor is a tumor, either it's benign or malignant. What does it matter if it's a carcinoma or a sarcoma? And the bottom line is there may be very different treatments, right? So these different types of cancer, depending on what cell type they originate in, may respond very differently to radiation or different chemotherapies. And so it's very important from a treatment standpoint to know exactly what you're dealing with. And so you don't have to memorize these, but I just wanted to point out that cancers are named based on the cell type of origin, so the location of the primary tumor. So for example, if you have a basal cell, if you have a benign tumor, it would be considered a basal cell adenoma. If you have a malignant tumor, it would be considered a basal cell carcinoma. Or like I mentioned, my wife's example, she had a meningioma. That's the meninges, or the cells that line the brain and the spinal column. So benign tumor would be a meningioma. If it's truly cancerous, it would be a meningiosarcoma, etc. So the names almost always indicate the site of the original tissue. And again, that's important for treatment purposes. All right, so we have these cells that can invade, that can metastasize, and we'll talk about that in more detail in a coming chapter. But the last part of this definition is that cancer makes us sick and can kill us. So unfortunately, we're all aware that cancer kills, but why is that? Why does it kill us? And there's really two main causes of cancer-related deaths. One is the tumor, the cancerous cells basically interrupt the function of some critical organ. So you're basically replacing the normally functioning cells in that organ with cancer cells, or the tumor just gets so large that it's blocking the function, the physical function of that organ. Or cancer may interfere with the immune system, and then that leaves you open to other potentially fatal secondary infections. So either of these could be huge problems. And as we saw earlier in the chapter, different cancers vary in their ability to cause death, or their likelihood of causing death. And oftentimes you'll see cancer death rates, mortality rates, expressed in terms of 5-year survival rates in many cases. And so these 5-year survival rates, was really kind of a commonly accepted metric in the field of cancer medicine and cancer research, is basically just looking at the percent of individuals that are alive at 5 years from the time of diagnosis. And we've seen the 5-year survival rate increase over the years. If we look in the 1970s at all cancer types, it was about a little under 50% were still alive at 5 years from diagnosis, whereas about 10 years ago, 10 to 15 years ago, we were at almost 70% survival. So that's a good increase. And you can see for many of these different cancer types, there have been various levels of increase in the survival percentage. Right, look at prostate cancer down here, from less than 70% to almost 100% at 5 years. So, again, just because you're alive at 5 years from the time of diagnosis doesn't mean that you won't eventually die from that particular cancer. Doesn't mean you're cured at 5 years, but it does mean that treatment has probably kept you alive and at least improved your quality of life. One possible skewing in the change of these numbers is what's referred to as this lead time bias, which is referring to the fact that as we develop more sensitive methods for detection, and we're able to detect cancers earlier, that can sort of artificially skew the 5-year survival numbers, right? Because if you're detecting it earlier, and that detection is where you're starting the clock, then of course 5 years out, even if the overall survival rates haven't changed, or the treatments haven't changed at all, your 5-year survival rates will likely look at least a little bit better. So I think it's important to think about, okay, 5-year survival is definitely a useful metric, but you really also need to pair that with overall mortality rates. So, what percentage of people at any time point are dying from these particular cancers? And so we've seen a lot of grim numbers here in this chapter so far, but I do want to show you some optimistic statistics here. So we'll talk about this over the course of the semester, but we've poured a massive amount of money and and human research hours and effort into trying to come up with better cancer treatments and diagnostics, etc. And so there have been some estimations of, well, how many lives have been saved, how many deaths have been averted due to all this effort over the years. And so if we look at the increase in cancer rates basically before much of this effort really started, um, and we just project that hypothetical line forward into the future, which would be the red here in both of these graphs for male and female, but then compare that to the actual lines, which have actually leveled out quite a bit, right? Basically everything between those two would theoretically be our number of deaths averted, and that's a pretty substantial number. So I think you could certainly argue that even though we haven't cured cancer, and that frustrates a lot of people, we do see more cures than we were seeing before, and we do see lots of treatments that are at the very least allowing people to live with their cancers for longer periods of time. And hopefully those numbers will continue to improve as technologies and medicines continues to improve as well. So I'm sure you've gotten the idea just in this first chapter, right? Not all tumors, not all cancers are created equal. And this is highlighted by the idea that every tumor, even if you're talking about, for example, let's say two different breast cancer tumors, every tumor gets graded based on their particular pathology. And so tumors get this grade of 1 through 4, where a higher number is worse, meaning a more severe tumor. And what are they looking at to try to determine that grade? Well, you can see some of those things in this table here. So look at the size of the nucleus in those cells. What's the ratio of the nuclear size to the volume of the cytoplasm? What's the shape of the nucleus? Basically, what's the number of dividing cells? What's the tissue organization look like? How differentiated are the borders of the tumor, the boundary, etc. So you would have to actually have somebody, or maybe even a computer, that's trained to identify those different variables and then give that individual tumor, based off of a biopsy sample, a particular grade. And then on top of the grades, we also have staging, and you've certainly heard of this. So it was a stage 3 cancer. And again, the higher the number, the worse the prognosis. And so there's a number of different staging systems that are used, but the TNM system is probably most widely used. And that stands for, the T is for tumor size, the N meaning has it spread to a lymph node, and then M has it metastasized, basically spurred secondary tumors in the body. So with the grading and the staging, you have some important information, and it's very useful, very important, because the grade and the stage can affect survival rate in a big way, right? So here we're looking at the 5-year survival percentage based on tumor grade or tumor stage. And if you're a patient, or for sure if you're an oncologist trying to treat a patient, you would want to know, okay, am I dealing with a stage 1, or am I dealing with a stage 3, or a grade 1 or grade 3, sorry. But yeah, depending on where you're at, right, that may also influence the treatment plan, the urgency, etc. And I should also point out here that, in addition to these kind of more traditional grading and stagings, a lot of modern cancer sort of diagnosis and prognostication comes from looking at differences in gene expression patterns in cancer cells. And we'll talk about this as we go through some other chapters, but this idea that changes in the expression of specific genes in a tumor may also be a very good indication of how severe this particular cancer might be, or how that cancer might respond to particular treatments. So that's kind of a more modern approach. but it is being used more and more frequently. So we've gone through this long definition, and hopefully that gives you at least the beginning of an appreciation for the complexity of what causes cancer. And again, we'll dive much deeper through the course of the semester, um, but I think it's fair to say that just saying, oh, it's just too many cells is sort of underrepresenting the problem. So we have this definition, but then that still begs the big question of, well, what causes cancer? And there's quite a few chapters in this book that deal with various causes of cancer. But in general, I think it's fair to say that it is ultimately mutations in genes, changes in particular genes in your cells that cause cancer. And we'll get into a lot more detail, but I did just up front want to introduce these two main sort of classes of cancer-causing genes, and they're what are called oncogenes or tumor suppressor genes. And these are genes that really have opposing biological function. So oncogenes are genes that promote cell division, encourage more cell proliferation, whereas tumor suppressor genes are those that would normally act to slow down or suppress cell division. So they have these competing actions. And you can imagine if you get oncogenes that become basically hyperactive, right? So they're promoting too much cell division, that's a problem. And similarly, if you have tumor suppressor genes that basically lose their ability to suppress proliferation, that's a problem. And in fact, what we see in most cancers is changes to both of these classes of genes. Yeah, we see oncogenes that are becoming hyperactive, and we see tumor suppressor genes that are becoming inactive or hypoactive. And the specific thing that's ultimately causing that, whether it's a carcinogen or a virus, we could say doesn't really matter. This is really the true mechanism, the ultimate cause of the particular cancer. But we will look at that in more detail. So coming up in chapter 2, we'll look at what they're calling the profile of a cancer cell. But what are some of these specific characteristics that cancer cells possess that make them cancerous, right? That really contribute to this disease pathology. So that's where we're headed in chapter 2.